Good morning, everyone. We will start with today's session. You know, the topic which is already displayed, demystifying the process of generic drug development. Before we begin the lecture of Mr. Mahesh, let me briefly introduce him. Mahesh Patil is an alumni of Department of Pharmaceutics of Guru Nanak College of Pharmacy. Very bright student. I have several formulations with him. He was the first person who started Jignasa and the working model, which is still continuing. So he was a student in charge at that time. Also, we had a quiz competition, and almost 300 students had participated in that quiz competition all over Maharashtra. It was also conducted by Mahesh. He has very good experience of research and development, especially in the generic product development. So now I hand over the charge to Mahesh. He will just introduce himself from his side and then we'll begin with his talk. There is a request. Please unmute yourself during the lecture time. If you have any questions, you will just use the raise hand option. Can you hear me?
Someone is unmuted, so that's why there is a noise.
We will just we'll hello.
Hi, madam. Yes, now your voice is very clear.
Yeah. So we'll start, sir.
Yes, yes, definitely. Uh, so first I will check audio video, then we'll start. Okay. So if my voice is very clear and loud to you, please type yes or Y in a chat box, so I can get how many of people can hear me. Just type yes. If my voice is clear to you and it is audible, then just type yes. If you have a problem, type no. So my audio is very clear to all, and I think I will start now. Uh, so before proceeding to today's topic, let me introduce myself. Very short introduction I will give you. Uh, Total, I have almost 8 years of experience, and uh, not all is in a R&D department. Let me clear, I have only 3 to 4 years of experience in research and development, including my post graduation also. So as a career path, my career path was very zigzag type of, you can say, as a Brownian motion. uh, I started my career in 2005 after completing diploma in pharmacy, and I joined USV Limited as a production officer. And then in a 1 year, I left the company, completed my graduation from Nagpur University, and then completed my post graduation. And further, I joined production department as an officer in Glenmark's Generics Limited. There I worked for 1.5 year. In between, I also worked in academic also for 1 year. Then further joined production department and work in a Zydus Cadila for 1 year. And then similar type of profile, I was work for Alembic. And since 2016, I am in a R&D department. Previously, I was in a R&D department of Copran Limited, and now I am in a R&D department of Bliss GVS Pharma Mumbai. So basically, I have thorough knowledge of production department and very less of R&D. and I make my career I choose my career because I have a production knowledge and somewhat of research knowledge and post graduation. That's why I choose process development rather than formulation development. So you can say I am an expert of what process development rather than formulation development. But uh, I am a good person to know you, to brief you the generic drug development process. The overall process you must know, but you must be expert in one area. Okay, so this is what my short introduction. I think it is sufficient. So can I start, madam? Uh, the process is what demystifying the topic is what demystifying the process of generic drug development, and my today's objective is to simplify you the overall process of generic drug development. And I will talk basically regarding the process of generic drug development for US and rest of the world market, not for a domestic Indian market. Okay, so for regulatory market, what is the process of generic drug development? That will I try to simplify. Okay, so let's begin. Generic versus branded. I this point I need to know from you that what is your understanding about the generic and branded. So anyone can brief me what is generic and what is branded. Yes, please, Labhesh. What is difference between generic and branded? What is your understanding regarding generic and branded? So it is important for me that what you know about the generic and branded. Then we will see the process of generic drug development. So anyone want to answer? Yes, Isha, please.
The generic drugs are the drugs which are made after the patent expires, and the branded drugs are the original drugs which come under the patent. These are the patent-proof drugs.
Yeah. Correct. Anything, anyone want to add to it? Okay. So generic and brand name medicines have the same active. I'm briefing you what is major difference between generic and branded, and what is the similarity between generic and branded. There is a misconception that generics are cheaper drugs and they have misbranded, means they have low quality. So it is absolutely wrong. The generic drugs are having a similar type of quality as of the branded. The price difference is because the generic drugs doesn't require much of scientific knowledge to gain. So the research cost of generic drug is very less compared to branded drug. Okay, and quality point of view, both are similar. Okay, so generic and branded both have same active ingredient, both have similar criteria for safety, both have similar effectiveness, both have similar strength, quality, benefits. Okay, but they may look different. Okay, so shape may be different, color may be different, packing may be different, but the quality, safety, efficacy, all are similar in generic and branded drugs. And the major difference that is a major objective for generic drug development is that the generic drugs are cost effective than the branded drugs. Why we are making a generic drugs? To have a cost effective medicines. Okay. The 20 years of patent period is given to the branded drugs because they can for this period they can um, collect their research costs, okay, from the customers. But after 20 years, everyone should get medicines at a cheaper rate. That's why the generic drugs are developed. The major objective is what? To develop similar quality product with lesser price. Okay, so that is the major objective of the generic drug. So we will see the process. So before uh, going to the next slides, I will brief you. You have here good manufacturing practices, good laboratory practices. So there is a good product development practices also for a generic drugs. So it is not, you will not find it in somewhere in literature even. It is not a regulatory guideline or something. It is just my thinking that what is a good development practices. If you going to develop a generic drug, so what good practices you should follow, that I have listed here. So if you want to minimize the failure of drug development, generic versus branded, if you want to minimize your failure chances, then you should follow these good development practices. If you follow these development practices, there will be little chances for failure. Okay, so choose from the same type of available excipients. Thumb rule one is what? You should choose the similar type of excipients. Means whatever the excipients used in a branded drug, that should you have to use it. Okay. So sometime what happens? We are trying to develop a generic drug from a branded drugs. We need to copy it. Okay, copy whatever it is bad or good or good. Don't try to use the. So the thumb rule of generic drug is you no need to take any additional say features to value addition to the product. If a branded drugs, you don't need to add a value addition. Choose the similar excipients, whatever listed in branded drug, you have to choose the similar excipients. If you move from similarity towards the dissimilarity, your chances, probability of failure will be increases. So this is the first rule of branded drug development. Okay. So choose why I am telling you the failure is not the product failure. If you develop a generic products and it is failed, it is not the product failure. It may be the failure of regulatory submission. So regulatory authorities will check all this. If you use something different excipients than the branded, they will ask you for justifications. And this is very powerful and very costly. So if you want to develop a generic product from branded and want to file it for a regulatory market, you need to follow these thumb rules. Okay. Choose from the same type of available process. Next point is what? Process need to be ensured that the similar type of process we should need to choose. If branded drug is manufactured by direct granulations, then you don't need to choose the weight granulation process. If you don't have this facility, then skip this product. So this is also a thumb rule. Don't deviate the process. Don't do the innovations. Okay. So next is the efficiency of generic product versus designated reference product must be proved through the bio study. So what our final objective must meet. It is our objective. Okay. So in that I want to add some informations to students that not all the countries require the bio studies. Okay, there is a guideline for a bio equivalence also, bio waiver also, but most of the countries don't require bio study. In this country, the generic drug development process is what you need to just copy. You need to show a multimedia dissolution profile, and that's it. So it is the rest of the world ROW countries. Most of the ROW countries doesn't require bio studies. Okay. Uh, Everyone is clear? Please type yes. So, madam, it is. I am going very fast, or it is okay?
It is okay.
It is
It
okay,
is
na?
okay. Yeah.
Okay. So the last point is facility and procedure used to manufacture generic product must meet the same CGMP requirement as the branded name product. So in this point, I want to add, if you develop a product in R&D and not consider your market, then it is a big failure. Okay, you develop a product for US market, and you want to manufacture and register it for a Canadian market, then there is a difference in CGMP requirements. So very little bit differences that you must be captured. Okay, so during what this thumb rule tells you, you need to study more and more before selecting your products. So you need to consider all these points at the time of your product selections. That's what I want to say uh, as a good product development practices. So, what happens previously, development was not regularized. Okay, there is a very little regulatory constraint towards the formulation development and research. Okay, nowadays development regulatory authorities are also asking for development data. How many trials you have taken, how many show me the data, show me the means the data integrity also coming into focus in a R&D department also. So previously what only QA is for a manufacturing, not for a development. So there is the eye of regulatory bodies to the development also. पे क्या था कि डेवलपमेंट आपने कैसे किया कोई पूछता नहीं था. So you just need to show data of bioavailability, bioequivalence. That's it. But now regulatory is asking you how trials you have taken, what is the rationale behind these trials, why you increase this MCC, why you decrease these. All these questions are regulatories are asking. Okay, so that's why the process of generic drug development becomes very critical nowadays. Okay, so developing a product is not a good not a big task. The developed product to file in a regulatory bodies, regulatory countries is a major task, and manufacture this in a commercial scale is also a big task. So developing is not that much a big task, but regulatory submission and queries to respond the queries and manufacture at a large scale is a big task. Okay, so whatever the next slides, it is based on whatever my understanding, and the thumb rules. Let me admit one person. Okay. So the overall presentations I have divided into two major sections. First, we will thoroughly go through these presentations, and I will brief you what is the process of generic product development. And in a second sessions, which will be going almost 45 minutes. In that, I will show you some case studies, how the R&D department is working, and what is our daily routines, how we will develop a process that I will explain you through product development reports. And in these first sessions, I have divided into three parts. One is what we actually do prior to development, means whatever the process is there before actual trials are taking place. Okay, so before taking actual development trials, there is a lot of process, and there is a lot of fact that you don't know, that I will explain you. Okay, and prior to development also, in the last sessions we will see the after development what are the bigger challenges, means scale up and validations and their requirement, all this. Okay, so the overall this presentation now and forth is divided into three major parts: prior to development, actual development, and post development. Okay, so prior to developments, there are three major key steps. One is product selection, second is preparing a design strategy notes, and third one is project scheduling. Okay, so all these three are very, very important tasks. The development is research, that much whatever the importance of research have, that much importance the product selection have. Okay, so we will see one by one. So first is the product selections. So what is the criteria for the product selections, and what are the various departments that are involved in product selections, that we will see. So uh, company is the business unit. Any research organization or any company is a business unit, and they are aim to have profit. Okay, the major aim of research is to have a profit, and to remain in a market is a major objective of any company for a development. Okay, so product development, the basic thing is what much amount of profit we will get. That is the basis for the product selection. But the similar types, we need to consider our facility, we need to consider regulatory requirements. If the cost and benefit ratio doesn't match, we will not select this product. Okay, so the product selection companies are thinking two ways. One way is a long-term research with long-term benefit, and short-term research with short-term benefit. Short-term research with short-term benefits have a shorter risk, and long-term research with big benefits have a bigger risk. And I want to add that only because of the reasons, I think in this pandemic we don't have medicines. In this pandemic we don't have medicines to treat this pandemic. Okay, so because most of the companies are thinking in a short-term research and short-term benefit and less risk than the long-term research and long-term benefit with bigger risk. Okay, so what are the departments that involved in product selection team? So actually, the product selection is done by a team, which is included the head of R&D department, head of regulatory department, head of legal department, head of marketing and sales, head of finance. Okay, all those heads are come together and discuss with the management body and select a product. Whatever the product selected for the research are done by input from these all departments. Okay, so everyone is clear up to this? Please type yes.
Sorry, sir, I'm not able to type.
Okay, let me. I can understand. Okay, so we will move to the next slides. Design strategy note. Okay, so it is a very important document. I think most of you have heard this word first time. So the company to company, these documents name is different. Okay, but the basis is what? You start the actual formulation development, you start actual R&D, you need to have a certain documents. On the basis of that, you will select the strategy. ओके, सो क्या भी जो भी आपको सिलेक्ट करना है, जो भी स्ट्रेटेजी आपको डिफाइन करना है एक्चुअल डेवलपमेंट की, वो बेस्ड ऑन दिस डिजाइन स्ट्रेटेजी नोट्स विल बी डन. Okay, so as a product selection team, they will prepare this document. Okay, so all the members of these product selection teams are responsible to prepare this document. What this document contain, the important information is I have listed by highlighted by bullet points. Innovator, product description and dosage form. You know the innovator? Yes, almost all will know the innovator. Innovator means what? The branded drug manufacturer is the innovator. Okay, so जिसको हमको कॉपी करना है, उसका प्रोडक्ट का डिस्क्रिप्शन चाहिए, उसका डोसेज फॉर्म चाहिए. सो ये बहुत ही इम्पोर्टेंट रहता है, बिकॉज as per my experience, most of the times what happens at the time of product selections, you uh, selected a product based on some different innovator, and in between your development process, there is a change in the innovator. So this is a bigger challenge at this point, the formulation development industry, the change of the innovator. Sometime what happens, innovator withdraw their products from the market. Okay, so you need to include the product innovator product description and dosage form into design strategy note, and you need to update this into your design strategy note. Okay, so innovator product packaging descriptions. So you can say the design strategy note is a continuous document. It need to be revised time to time, and all the concerned persons from the selection teams are liable to update this design strategy note. Okay, so innovator product sales that need to be included in design strategy note. Generic product descriptions, what generic products we are going to manufacture, and that description, tentative description you need to add, tentative packaging of your generic product you need to add into this, and what is the sell forecast that you need to ask. So all these points are required to be incorporated into a certain documents. For this company, it is design strategy notes. For other companies, maybe the name may be different. Okay. The third, the major important point is project scheduling. Okay, the scheduling is why important, because whatever the sale forecast you have done and whatever cost benefit ratio you have determined for a generic development, that is very badly impacted with your delivery. Okay, so I'm taking a 1 minute. I just going to check who is unmuted. Satyam Hatwar. Lavesh, please.
Sir, I am muted.
Okay. The third point is project scheduling. So it is very important point that need to be considered prior to the development. Okay. So whatever the sell, whatever the cost and benefit ratio generic development is very impacting factor is your project delivery. Okay, so developing a good uh, defined time frame is a bigger challenge to today's scenario. So there is a schedule, there is a project planning, and up to this planning, you need to deliver your product. And if you develop, if you develop and if you deliver the product after the timeline, it has no value. Okay, so that's why it is important. So what need to consider for defining the project schedule is patent expiry. When the patent is going to expire. ऐसा नहीं है कि पेटेंट एक्सपायर होने वाला है और हम डेवलपमेंट बाद में स्टार्ट करें. वी फॉर बिफोर वी विल स्टार्ट, वी हैव टू स्टार्ट द डेवलपमेंट प्रोसेस. Okay, product exclusivity need to be considered, forecast need to be considered. If we have a bigger forecast, we have a bigger profit, then we need to deliver the product in a short time. Then based on that, you need to define your project schedule. Okay, availability of active ingredients. Availability of active ingredients means what? Sometimes what happens, the active ingredient manufacturers is under development. So he's also doing a research and he's filing a DMF, and after that you will get the API, and then you will start the then your project will define based on that. Okay, so not only project scheduling is important, to be in line with this project schedule is also important. Okay, the time to time you need to revise these project schedules, and you need to be strictly adhered to the project schedule. So here, as an industry person, I want to add one thing. Want to told you students that there is a one department called project management. Okay, project man. You know that project management, guys. You know project management as a department. How many of you knows project management as a department? Type yes, type no. किसी को पता है प्रोजेक्ट मैनेजमेंट क्या होता है? Okay. I will explain you. This in a recent year, in the recent time, the project management is getting very good scope. If you, hello, yes, anyone want to add?
Please unmute yourself. Mute yourself, please. किसी ने तो भी माइक म्यूट नहीं किया है.
हाँ, yes. But we can manage it. Okay, madam. Slides are visible, na?
हाँ ठीक है, था कंटिन्यू कर.
Okay, continue. So project management is a separate department in R&D. They are directly reporting to the management, and they are actively involved in this project scheduling. Okay, so it is a good department, I think. For that, you need to have any management degree or a postgraduate is good. If you know the overall research, how the research is going on, and what are the major challenges to the research, and you know requirement of this research, then and management principles, then you are the good persons for the project management team. Okay, so for that you need at least 2-3 years of experience in R&D in any department. You need you need to know the overall process of research. Okay, so it is a good department, I think. You can consider as a career opportunity. So prior to development is completed here, now the actual development started. Okay, so before starting the actual trials, you need to do some literature survey. You need to evaluate your API. You need to evaluate your reference products. Okay, so that is called a pre-formulations. Most of the students consider the pre-formulation means what? Pre-formulation means drug excipients compatibility karna. Okay, so that's not only the pre-formulations. The pre-formulation is little bit more than that. Okay, so here in a pre-formulation, the QVD comes into the focus. The first time points where the QVD come into the focus is the pre-formulations. So literature survey is the first point. This is not the pre-formulation, I think. The literature survey is a continuous process, and it should start at the time of pre-formulation but continue till the product is in the market. Okay, so literature survey is a continuous process. We should consider, don't consider it as a pre-formulation. We will see one by one. Please give me a 1 minute. Okay. So for literature survey, what you need to require, what you need to search. Okay. So first is you need to obtain and review your product selection document. That I told you, design strategy note, which is prepared after the product selections. That you need to review, you need to understand these product selection documents. Okay. Second is you need to review and obtain your patent information, your project related patent information. There is a separate department of liaison department which will give you summary review of your patent survey, patent literature survey, and that you need to understand, you need to learn it. Okay. As a development scientist, maybe as a formulator scientist, maybe as a process development scientist, maybe as an analytical development scientist, you need to perform, you need to read these documents. Okay. One is product selection document, one is patent information. And the branded products, there are publicly available informations are available. Some are US FDA itself publishing product related guidelines. Product specific guidelines are published by US departments and other regulatory bodies also, that you need to understand. Okay. Merck index is there, and a lot of research articles, research papers. You need to collect all these information and you need to understand all this. Okay. So it is very important part of formulation development, I think. And it should start after getting product selection document, and it should last up to product submissions or up to the commercializations. Okay. Second point is reference product evaluations. Means what? Branded product evaluation. In this step, you need to evaluate your branded products. What you need to evaluate? You need to evaluate its packing, its overall physical appearance, its overall chemical performance, means dissolution, assay, all these points, chemical evaluation you need to perform. Physical evaluation like shape, size, thickness, hardness, this you need to evaluate. You need to evaluate packaging of your reference product. Also, you need to evaluate the stability of your reference product. Uh, Sometime what happens, your reference product itself fails in accelerated stability studies. So you need to perform the stability of your reference product also. Okay. Uh, so what is your T0 points? Means from where actual development starts. So the actual development starts once you get both your reference product and your API. All are getting my points. So I'm feeling it is one way. Hello, madam.
If we go for two-way, there's a lot of noise. So just go on.
Yes, yes. No, actually, it's the first time. It is a. I don't have a experience of this virtuality. So please, at least you disturb me in between the presentation. So I will get. Yes.
I will just request students, if they want to ask any question, please feel free to ask, and they will just raise their hand and ask the question.
Yes, if any technical issues are there, we can get this. Okay. If I'm too much fast, or it is okay, madam?
No, it's okay. Just go on.
Okay. So you got the points. What reference product you need to evaluate? Okay. So what I'm discussing, telling you that what is T0 means when actual formulation development will start. means now your time start, you start to develop, is the point when you get your API and reference product, means branded product. Okay, so once you got the API, once you got the branded products, your time starts. And before that, you can do the literature survey. You can basis of the literature survey, you can arrange the required excipients, you can arrange the required other resources like equipment for analysis purpose, columns and all this. You can buy this. Okay, so it will not impact further to the your projects. So third is drug substance evaluation, means API evaluations. So it is very important points you need to consider for the good development. Okay, so what you need to evaluate for your API. Okay, first point is polymorphisms. Whatever the API you got, you must need to determine the polymorphisms. What happens? The API that used in a reference product may be manufactured by the different route of synthesis, and your API may be manufactured by different route of synthesis. That will impact your polymorphic form, that will impact your residual solvent, that will impact your related substances, that will impact your impurities. All these are badly impacted. So you need to consider all these points, and you need to evaluate your API by considering these points. ओके, पॉलीमॉर्फिज्म आपको चेक करना है, ड्रग की सॉल्युबिलिटी आपको चेक करना है. इफ द एपीआई route of API synthesis is changed or anything is changed, then solubility will also going to change. You need to determine the solubility of your drug substance, pH dependent solubility of your drug substance. Its particle size, which is very, very important. So whatever the API you got, you need to check the particle size. You need to check its flowability. Chemistry related points like your related substance, solvent, residual solvent, all these. Okay, and incompatibility like sensitivity to light, sensitivity to heat, sensitivity to moistures, all these you need to consider for a drug substance evaluations. So it is a very, very long process. You need to and you need to perform it throughout your development. Okay, see the particle size. If your product is a BCS class 1, highly soluble, highly permeable, then you don't need to worry about the particle size. Okay, so वो आप अगर बाद में भी आपने आइडेंटिफाई किया, QVD में भी आपने चेक किया तो भी चलेगा, लेकिन ये सब आपको करना ही पड़ता है. So why? Because it is regulatory compulsion. Okay, if you think your particle size of your API doesn't impacting your formulation, then you need to justify it. Okay, so you need to perform any means risk assessment, or you need to perform any trials. On the basis of that, you need to give regulatory that the particle size is not impacted. So a lot of queries I have seen because of the particle size of API is not done, and a total formulation is completed. Okay. So the next, after the pre-formulations, you will start for the actual process of development trials. Okay, now you can say there is start of your trials are, you are now able to start the actual development trials. But before that, I want to add one more points to these 3 points. Okay, the literature survey हो गया, reference product evaluation हो गया, drug substance evaluation हो गया. After that, as a part of pre-formulations, I want to add here QVD. Okay, actual quality by designs come into focus at this point. At this point, after evaluation, based on your reference product evaluation, based on your drug substance evaluations, and based on the document you got from the product selection, you need to define your quality target product profiles. and based on that quality target product profiles, you need to perform your further evaluation, further development trials. So at this point, you need to do a risk assessment. What risk assessment you need to perform, what are the QTPPs, that all I will cover in a case study sessions. Okay. So next is the process selections. Process selections, the basis of process selection is, first is what, you need to check for the process that innovator has choose. Okay, innovator का जो भी process है वही process आपको select करना है. Unless and otherwise, if there is any concern of availability of equipment or availability of facility or cost issue, you need to select the same process as that of the innovator. Okay, अगर कुछ concern है तो ही आपको method अलग choose करना है. Okay, so you can directly start with the reference of your literature that innovator has followed this weight granulation method. So I have selected the weight granulation method. You can write directly this. Okay. Otherwise, if there is you doesn't got any information about the innovator, which method innovator has used, that time you need to select your method. The first choice is a dry mix, direct compression. Second is dry granulation, and then wet granulation. Why? Because the dry mixing is, very simple process, very less variabilities, and very cost effective. Require the less excipients, require the less process issues. That's why it is first choice of method. Okay. So the next task is to select the excipients. What you need to consider for selecting the excipient. First, I told you the thumb rule is what, you need to select the excipient that innovator has used. Okay, in a literature survey, you can get what excipients innovator has used, and you have to select the same. Okay, so in this point, I'm going to add you the one term, reverse engineering. Okay, so reverse engineering is what, in that your innovator, you will check the excipients quantitatively by reverse engineering. Okay, reverse engineering is a process by which you can check the excipients quantity in a brand product. Okay, so by chemical analysis, you will get how much approximate amount of XYZ excipient is used in a branded product. Okay, अगर branded product is BCS class 2, and then by literature you come to know that it contains SLS, then you need to check the quantity of SLS. Okay, so it is very critical for you. So that's why for selected excipients, the reverse engineering is done. So you will get the exact amount, approximate amount of excipients. Okay. So the first selection thumb rule is what, you need to follow the innovator, and if it is not possible to follow the innovator, then you need to choose the excipients. So I paused. I am admitting one person, Harshali Shende. Okay, let me make her as an attendee. So please give me a 1 minute. Okay, So first point is composition of reference product. On the basis of reference product composition, you need to select the excipients. This is about the excipient selected. What about the grades? The grades is selected based on your QTPP documents. ओके क्वालिटी टारगेट प्रोडक्ट प्रोफाइल्स होगा, उसके बेसिस पे आप क्या करेंगे कि व्हाट इज योर रिक्वायरमेंट, हाउ फास्ट you need to release your drug. Okay, so based on that the grades is selected. If it is a binder, if it is a release retardant, then the grade is selected based on this. Okay, so requirement for the specific excipients is also come into focus after QTPP. Okay, drug excipients compatibility. In that, from the initial literatures, you will get the idea this drug is not compatible with these excipients. Okay, so after selecting the excipients, also you need to perform the drug excipients compatibility before starting the trials. This will screen your excipients list. Okay, if initially you choose the 15 excipients, maybe 2-3 are incompatible, you will get the another one. Okay, the selected process, based on the process you have selected, if innovator have selected the weight granulation, and if you think I am taking a risk and I am going to develop a product with direct compression, then your excipients may be different than the excipient than the innovator. So formulation process is also impacting factor for the excipient selections. Further, the availability. So excipients must be readily available, and based on that you need to select. Okay, so here for the availability, I want to add one. I want to share my one experience with you. During the development of one formula, it's amoxiclav dry syrup. They have used succinic acid. Okay, succinic acid is used by the innovator, and I have stuck almost for 1 year to get the succinic acid. Okay, domestically the succinic acid is not manufactured, and the succinic acid from the China and from US, UK, to get the samples of succinic acid, the almost 6 to 7 months of development period was spent, was on hold because of the availability of succinic acid. So what happens? The innovator is manufacturing in a US, and the succinic acid is available in a US, readily available. So that's why he used the succinic acid as an pH stabilizer. So it is not available in India, and I want to develop in India. Then I need to go for the other. Like then finally I have replaced the succinic acid with citric acid and stabilize the formula. Okay, so this is what I'm going to say. The excipient selection is thumb rule is what the branded product, but it is a thumb rule. It is a so first choice, but there is a cases, there is a strategy tragedies by which the excipients may be different in a branded and generic product. Okay, third is experience. Experience means what? You have the experience, you have developed one extended release formulations, and you have used HPMC, and you have good knowledge, good experience of how much amount of HPMC is required to retard the release of such a kind of products, such a kind of API, then you can use the similar excipients. Okay, so this is also the impacting factor for excipient selection. The last one is the cost. See, the cost is at this point, the cost, it is better not to select the cost. Okay, at this point you need to select the risk of product failure. Okay, so cost को अभी consider करना, I think it is bad for a initial point. Initial point में आपको क्या करना है कि risk को consider करना है. So cost अगर है तो हम we can develop alternate vendor and you can reduce the excipient cost. But for selecting the excipients for R&D purpose and considering the cost is limiting your ability to develop a good formula. Okay, so as a formulator, I want to add what you need is a wide vision. Okay, what you need a wide vision. आपको है ना लिमिटेड थिंक नहीं करना है, अभी फिलहाल के लिए नहीं सोचना है, लॉन्ग टर्म सोचना है कि अभी आपने अगर आप खुद को लिमिट कर लिया सो आप अच्छा फार्मूला नहीं बना सकते. ओके, तो ये लिमिटेशन नहीं रखना रहता है एक्सिपिएंट सिलेक्शन का कि कॉस्ट इज विल नॉट द क्राइटेरिया फॉर एक्सिपिएंट सिलेक्शन. ओके, so up to this point it is clear?
Yes, it is very clear.
Okay, okay. So the next slide is the formulation development. Very frank, you consider the formulation development or actual research at this point. Okay, so this point from most of the majority of the students' point of view is a formulation development or it is a research. So before this, I have almost covered 15 slides, which is prior to the development. So it is a part of research and development, and it has to be very seriously taken into considerations. So actual trials जो ले रहे हैं उससे ज़्यादा importance होता है जो previous slides में मैंने explain किया. So that's all must be thoroughly studied, must be considered, and must be incorporated into the risk of development failure. अगर कोई डेवलप प्रोडक्ट अगर फेल हो रहा है तो उसका रीजन आपका ट्रायल्स नहीं है, और मे भी आपका प्रोडक्ट सिलेक्शन हो सकता है, मे भी आपका लिटरेचर सर्वे हो सकता है. सो what today's scenario is what systematic approach, systematic risk-based approach in formulation development is today's scenario. That's why the QBD is come into the focus. Okay, so actual formulation development is what we will start here, start taking the different trials. So you might have a question that पहला ट्रायल कैसे चालू करेंगे, कितना क्वांटिटी लेंगे. So the basis for the first trial, the composition of first trial is based on prior scientific knowledge. Okay, तो prior scientific knowledge पे आप एक formulation start करते हो, उसके बाद में आप छोटे मोटे ऐसे 15, 20, 30 ट्रायल्स लेने के बाद में आप एक टेंटेटिव फार्मूले पे आते हो और आपका प्रोडक्ट नॉलेज जो है वो थोड़ा डेफिनेट हो जाता है कि आपका प्रोडक्ट अंडरस्टैंडिंग थोड़ा हो जाता है. उसके बाद में हम क्या करते हैं कि डिफरेंट एस्पेक्ट ऑफ फार्मूलेशन आर स्टडीड बाय QBD. Up to this point, initially to apply the quality by design and to apply design of experiment is not good. You need to apply the design of experiment after getting certain initial knowledge from initial trials. Okay, so पहले जो 2-4 ट्रायल्स आपको लेने के हैं उसमें आपको गेन होगा कि हाउ द प्रोडक्ट इज बिहेविंग, हाउ द your generic product, how your test product releases, and how your branded product releases. Okay, so based on that, next task is what you need to perform design of experiment. You need to check impact of your particle size on your dissolution, impact of your excipients grade on dissolutions, based impact of your excipients grades on stability. You need to check the stability. All these come after this. Okay, so initially you need to take 4-5 trials based on prior scientific knowledge. पहले जो डेवलपमेंट हुआ है उसके बेस पे आपको एक टेंटेटिव फार्मूला पे आना है और उसके बेस वहां से आपको स्टार्ट करना है. ओके, सो व्हाट you need to check of developed product. So as it is very simple, जो जैसा हम एकेडमिक में करते हैं वैसा ही सब कुछ चीज़ें चेक करते हैं. एकेडमिक में सम टाइम हम स्टेबिलिटी नहीं चेक करते हैं, यहाँ पे हम स्टेबिलिटी चेक करते हैं. तो क्या क्या चेक करना है आपको, ब्लेंड अगर है तो उसका BD TD चेक करना है, पार्टिकल साइज डिस्ट्रीब्यूशन चेक करना है, फ्लो चेक करना है, एंगल ऑफ रिपोज चेक करना है. So as a good formulator, the good formulator is who can generate more data in less number of trials. is a good formulator. It is a thumb rule of formulator. If you are interested in a formulation development department, so take it as a Brid Vakya, generate more and more data, record more and more. Okay, so recording more and more will get a more data and will get a more conclusion. Okay, and it will help you to better understanding your product. So formulator is a father and who is going to give birth to the new child, and he should know each and every part of his child. Okay, तो उसके बारे में थ्रू आउट अगर आपको अंडरस्टैंड करना है तो आपको मोर एंड मोर डाटा आपको जनरेट करना है. सो सिर्फ डाटा जनरेट इज नॉट योर एम. देयर इज लिटिल बिट बियॉन्ड गाइडलाइंस. गाइडलाइंस ऐसा नहीं बताएंगे कि आप अच्छे से ऑब्जर्व करो. सो ऑब्जर्वेशन इज आल्सो गुड मींस रिक्वायर्ड टास्क दैट यू नीड टू दैट formulator must have. Formulator must have wide vision. Okay, if the objective of the experiment is to check the dissolution profile and compare the dissolution profile with the branded drug, then your focus, all the focus must be in a dissolution, but the same time you must have the wide vision to check other parameters also. So for a tablet you need to check appearance, average weight, weight variation. All these are the normals, and all you know this. Okay, so whatever the development goes on, that is recorded as a process, that is recorded in a laboratory notebook. Okay, and nowadays most of the companies are using digital laboratory notebooks, and I'm very surprised with Bliss GVS. It is not a big company. My company is not a big company, but we are using digital laboratory notebooks. Okay, some of scientists have a tablet and they are recording the data online in a software Chemia. Software name is Chemia. So all the development data is recorded. कितना RPM रखा था इम्पेलर का, कितना mg लिया था वो फार्मूला शीट, और क्या LOD आया था, कितना मिनट ड्राइंग किया था, वो सब ऑनलाइन रिकॉर्ड होता है इन टैबलेट और इन लैपटॉप पीसी में ऑन अ सॉफ्टवेयर. ओके, सो डॉक्यूमेंट नाम का कोई चीज नहीं है. व्हाई इट इज इम्पोर्टेंट एंड व्हाई nowadays R&D is also following this electronics BMR, like electronics LNB, because regulatory are asking for the data. Okay, तो क्या हो गया है कि डाटा इंटीग्रिटी इशू इन अ फ्यूचर टाइम विल डेफिनेटली कम इनटू फोकस फॉर द R&D आल्सो. ओके, R&D आपने कैसे भी करके एक फार्मूला बनाया और उसको आपने फॉर दिस टाइम पॉइंट बायो इक्विवेलेंट अगर कर दिया, इट डजंट मीन यू हैव डेवलप योर प्रोडक्ट. Need to have the scientific understanding of your critical material attributes and your critical quality attributes. Okay, जो भी डिसोल्यूशन है, जो भी रिलेटेड सब्सटेंस है उसका, और आपके जो फार्मूला में जो एक्सिपिएंट्स है उसका, मटेरियल का प्रॉपर्टी, वो सब लिंक करके आपको एक उसका रिलेशन एस्टेब्लिश करना होता है. तो वो रिलेशन रेगुलेटरी बॉडीज नाउअडेज आस्किंग. So that's why these electronics laboratory notebooks come into focus. और उसमें ऐसा नहीं है कि आप फिर दूसरे दिन आके उसमें डाटा रिकॉर्ड करेंगे और कुछ भी आके कुछ भी नहीं भी आया था, LOD इतना नहीं था तो भी आप लिखोगे, ऐसा नहीं चलेगा. तो thoroughly as per design of experiment, the formula sheet goes into the laboratory notebook. To the particular space you have to add the remark. In a remark column you have to add the whatever the observations you need to put into your LNB. So it is 21 CFR, that's why you cannot modify or manipulate it. Okay, so that's is what Bliss GVA is following. So moving to the next slide. Okay. So क्या होगा? Yeah, is up to this point I explained you what we did. Initially we did 3-4 trials and come to the approximate formula, and then based on QTPP, based on product knowledge, we decide to take the QBD trials. Okay, अगर हमको ऐसा लगा कि yes, the particle size of API is impacting my dissolutions, as the dissolution is very slow in the initial time points, then हम क्या करेंगे कि we will decide the strategy and we will take the DOE. Okay, so DOE के बेस पे फिर ट्रायल्स होंगे. Okay, सो पहले जो डेवलपमेंट ऐसे ही डेवलपमेंट कंटिन्यूज रहेगा, और हम कब बोलेंगे कि फार्मूला, ओके, फार्मूला इज फ्रीज कब बोलेंगे? व्हेन द फार्मूला इज मैचिंग द डिसोल्यूशन प्रोफाइल विद द ब्रांडेड ड्रग्स इन मल्टीमीडिया. ओके, 0.1 नॉर्मल HCl में होना चाहिए, एसिडिक मीडिया में होना चाहिए, बेसिक मीडिया में होना चाहिए, फॉस्फेट बफर में होना चाहिए. वो सब मीडिया में अगर मैच हो रहा है डिसोल्यूशन, एंड वी हैव इनफ कॉन्फिडेंस दैट now there is a less chances of bioequivalence failure, and then we will put these batches, larger scale batches, into a stability. Okay, so as an project schedule, I want to tell you that what is time frame. क्या बेसिकली क्या टाइम फ्रेम रहता है और कैसे टाइम फ्रेम में इंडस्ट्री काम करता है वो मैं बताता हूँ. कि T0, I told you, T0 start when you got your API and excipients, API and your reference product. Okay, once you got your reference product API, then your T0 starts. Now based on the complexity of your product, you will get 6 month or 3 month. Okay, so 6 month or 3 month में you need to develop, you need to freeze your formula. Okay, सो ये 6 महीने के पीरियड में आपको इनिशियल ट्रायल्स लेना है, क्यूबीडी लेना है, आपको फार्म जो आपका क्रिटिकल मटेरियल एट्रीब्यूट्स है और क्रिटिकल क्वालिटी एट्रीब्यूट्स है उसके बीच में रिलेशन एस्टैब्लिश करना है, आपको डिज़ाइन स्पेस एस्टैब्लिश करना है. ये सब आपको उस टाइम फ्रेम में करना है. ओके, तो उसके बाद में यू विल गेट additional 6 months for the stability, 3 to 6 months for the stability. Okay, stability means what? Accelerated laboratory stability you need to check कि प्रोडक्ट फेल हो रहा है, हाउ इट इज बिहेविंग, स्टेबिलिटी रिलेटेड सब इम्प्योरिटीज बढ़ रही है कि नहीं, आशा इज डिक्लाइनिंग और व्हाट इज एनी फिजिकल चेंज इज देयर और नॉट. So all these are parameter are checked in development stability, and once the development stability is okay, then after a 6 month, if up to the 6 month the data is okay, then the formula
महेश आवाज गेला.
now it is okay.
Yes, it is okay.
Yeah, okay. So can I repeat the last point?
Yes.
Yes. Okay, so the 6 month you got you will get for a development stability, and after completion of development stability you need to scale up your product to the pilot scale. Here I want to add certain products required pilot bio study, and certain products may not require the pilot bio study. And certain products even don't require the bio study. Okay, there are certain is in rest of the world ROW market where you don't need to have bioavailability data. So in that in this country directly we can proceed for or based on 6 month data of 2 batches, 2 R&D batches and 1 large scale batch, commercial batch, you can submit your dossier. Okay, in the rest of the rest of the countries, regulatory countries, Canada, America, US, sorry, US, UK, Europe, rest of the Europe, Middle East, all these countries require bio study. In that, you need to submit two uh, um, pilot scale batches data, stability data, and bioavailability, and one small scale data, batch data. Okay, तो 3 बैच का स्टेबिलिटी डाटा अप टू 6 मंथ यू नीड, एंड यू नीड अ बायो स्टडी डाटा. ओके, तो क्या होता है कि बेस्ड ऑन कॉम्प्लेक्सिटी ऑफ योर फार्मूला एंड रिस्क, कि आपको अगर फार्मूलेटर को लगता है कि देर इज अ रिस्क, प्रोडक्ट इज बिहेविंग वेरी डिफरेंट, and maybe there is a possibility of bio study failure, then initially for a pilot batches, bio studies check on a very small number of users. Okay, small number of bio members, and then after getting confidence, product is scaled to the exhibit scale, and on a exhibit batches the bio study is performed, and similar exhibit batches the stability study is performed. Okay, तो क्या होता है कि ड्यूरिंग द डेवलपमेंट यू गॉट द 6 month, in that you need to perform initial stability also. The stability is very important, and if you got the, because why I'm saying the stability is important, because you get the result after 6 month. क्या होने वाला है वो आपको 6 महीने बाद में पता चलने वाला है, बराबर. सो वो 6 महीना आप वापस नहीं ला सकते. उतने 6 महीने आप जो कुछ नहीं किया है, वो अगर आपको फुलफिल करना है तो यू नीड टू स्टार्ट योर स्टेबिलिटी आफ्टर इनिशियल 1-2 ट्रायल्स. अगर स्टार्ट कर दिया तभी आपको 1-2 बैच आपको स्टेबिलिटी चार्ज कर देना है. सो स्टेबिलिटी, एक्सेलरेटेड स्टेबिलिटी शुड स्टार्ट आफ्टर गेटिंग API एक्सपीरियंस इन 1 month. योर टारगेट शुड बी you will start the stability in a 1 month. Okay, so it will further reduce your risk of timeline failure. So moving to the next slide, what happens after development? You have freeze your formula, you have scaled your formula to the exhibit scale, you have manufactured 3 exhibit batches, you perform bioavailability study, and see, I want to add here one point. Once the formula is freezed, you need to take 3 development batches, 3 exhibit scale batches, and 3 exhibit scale batches need to be charged on stability. And only after getting at least 3 months stability data, you cannot perform the bio study. It is a compulsory requirement of most of the countries. Like it is a requirement for the US and Canada also. And in Europe, certain countries ask for initial exhibit batches data, stability data, because what bio study is going to perform on a human, and your formula must be manufactured in GMP, and this formula must be stable at least for a 3 month. Okay, सो 3 month एक्सीलरेटेड डाटा आपको चाहिए स्टेबिलिटी का, बाद में ही आप बायोएबिलिटी को वो सैंपल भेज सकते हो. बायो स्टडी अगर आपको करना है, सो हियर इज आल्सो वन ट्रिक. यू मस्ट नीड टू perform bio study on throughout on similar lot of your reference product. See what happens, sometimes अगर पायलट में बायो किया है और पायलट में बायो पास हो जाता है और एग्जिबिट में बायो करने जाते हैं तो एग्जिबिट में बायो फेल हो जाता है. एंड और हमको क्या लगता है कि कुछ तो भी चेंज हो गया, इसकी वजह से वो फेल हो गया. पर क्या होता है, कभी कभी जो लॉट ऑफ ब्रांडेड ड्रग हमने पायलट बायो के लिए लिया है और लॉट वही बैच का ब्रांडेड ड्रग हमको एग्जिबिट बायो के लिए भी लेना है, तो ये जनरली इग्नोर कर देते हैं बहुत सारी कंपनीज और फेल्योर. ओके, So what happens if you found that your product is not bioequivalent? Okay, अगर bioequivalent नहीं है तो हम क्या करेंगे कि हमको फिर से आपको इसको रीडेवलपमेंट में लेना है। अभी अगर यहाँ पे आपका नॉन बायो इक्विवेलेंट हो जाता है तो क्या क्या आपका कॉस्ट जाता है, लेट मी एक्सप्लेन यू। फर्स्ट, फ्रॉम प्रोडक्ट सिलेक्शन टू एग्जिबिट स्केल मैन्युफैक्चरिंग, एनालिसिस से लेके मैन पावर कॉस्ट, योर फैसिलिटी कॉस्ट, वो सब कॉस्ट आपका इंक्योर होता है। सो इट इज़ अ वेरी वेरी बिग रिस्क that if your product is found non-bioequivalent after exhibit batches. Okay, तो इसी रिस्क को रिड्यूस करने के लिए आपको एक स्ट्रक्चर्ड अप्रोच आपको लेना रहता है। दो ट्रायल्स के बाद में आप फिर से बायो स्टडी भी कर सकते हैं। ओके, और फिर से आपको मतलब बायो इक्विवेलेंट भी बना सकते हैं उसको। तो वो कैसे, क्यों, दिस इज द मैजिक ऑफ डिजाइन स्पेस दैट नीड टू बी अंडरस्टैंड। हम क्या समझते हैं कि डिजाइन स्पेस, अभी फॉर एग्जांपल अगर HPMC है, HPMC फार्मूला में तो फ्रीज हो गया है। से फॉर एग्जांपल आई एम यूजिंग 5 mg ऑफ HPMC in 5 mg of HPMC in a formula, okay, and it is fixed. Then why I need to check with 2 mg of HPMC and 10 mg of maximum 10 mg of HPMC? I need to check whether what is the impact of change in the HPMC with the dissolutions and the change in the dissolution with the bioavailability. अगर ये आपको पता चल जाता है तो इसका बेनिफिट ये होता है कि नॉन बायो इक्विवेलेंट होने के बाद में आपको बहुत जल्दी प्रोडक्ट अंडरस्टैंडिंग मिल जाता है कि बायो इक्विवेलेंट क्यों फेल्योर हुआ, कि क्या मेरा गलती हो गया, मेरा डिसोल्यूशन प्रोफाइल मैच हो रहा है पर बायो मैच नहीं हो रहा है, तो व्हाट इज द रीजन। तो ये सब चीजें आपको पॉइंट आउट हो जाती है इफ यू फॉलोड स्ट्रक्चर्ड अप्रोच इन योर डेवलपमेंट। ओके, फर्दर और एक पॉइंट ऐसा है कि क्यों हमको QVD में जाना है, क्यों हमको स्ट्रक्चर्ड अप्रोच लेना है, कि बायोएबिलिटी ओके है, एग्जिबिट स्केल ओके हो गया, प्रोडक्ट डोजियर फाइल हो गया, और कमर्शियल करने को हम जाते हैं। ओके, आई विल कंसिडर दिस आल्सो इन पोस्ट डेवलपमेंट स्लाइड आल्सो। सो अगर हम कमर्शियल में जाते हैं और हमको लगता है कि अभी हमको सम एक्सपीरियंस आई नीड टू बी चेंज, something I need to be changed post scale up, okay, post approval scale up changes, सुपैक जो जिसको बोलते हैं हम, वो अगर आपको करना है तो उसके लिए आपका डिजाइन स्पेस अगर है आपके पास, सो यू कैन गिव जस्टिफिकेशंस एंड इट विल सेव यूर बायो स्टडी। अगर स्केल अप में भी बायो स्टडी करना पड़ता है, अगर आपको आपका प्रोडक्ट आपने एग्जिबिट स्केल तक अच्छे से मैन्युफैक्चर कर दिया, बायो इक्विवेलेंट हो गया, स्टेबिलिटी डाटा भी पास हो गया, उसके बाद में आप कमर्शियल करने को जब जाते हैं, डोजियर ओके हो गया, यू गॉट द ANDA एंड यू आर गोइंग टू स्केल अप योर फार्मूला टू द कमर्शियल स्केल। ओके, तो अब तब अगर उस टाइम पे अगर आपको आपका प्रोडक्ट अगर चल नहीं रहा है तो ये बिग रिस्क हो जाता है। ओके, तो रिस्क सिनारियो है, रिस्क मीन्स व्हाट, रिस्क टू द कंपनीज रेपुटेशन, रिस्क टू द कंपनीज रेवेन्यू। ओके, जो पैसा कंपनी ने रिसर्च में डाला है वो रिस्क में आ जाता है। तो रिस्क जो है वो बढ़ते जाता है। इनिशियली रिस्क कम रहेगा, डेवलपमेंट ट्रायल जब चालू होगा तब रिस्क कम रहेगा। अगर आपका एग्जिबिट तक चला गया तो रिस्क थोड़ा सा और बढ़ जाएगा। अगर एग्जिबिट होने के बाद में कमर्शियलाइज के वक्त अगर आपका प्रोडक्ट फेल्योर आता है तो ये बहुत बड़ा रिस्क हो जाता है। ओके, अगर अप्रूवल हो गया है और प्रोडक्ट चल नहीं रहा है, ये बड़ा रिस्क हो जाता है। तो उस टाइम पे रेगुलेटरी को आपको जस्टिफाई करना होता है सर्टेन पॉइंट्स। ओके, अगर तब आपको लग रहा है कि आपको HPMC थोड़ा सा बढ़ाना है, तो अगर आपने पहले बायो डिज़ाइन स्पेस एस्टेब्लिश किया है और आपके पास थोरो साइंटिफिक नॉलेज है, जिस पे आप बता सकते हैं कि भाई इनिशियली हमने चेक कर लिया था, ये इतना इम्पैक्टिंग नहीं है और ये सिग्निफिकेंट नहीं है, सो वी कैन इम्प्रूव इट, वी कैन इंक्रीज इट।
and it is not going to impact your bioequivalence. Okay, तो ये चीज इसीलिए इतना ज़्यादा इम्पोर्टेंस है। रिस्क बेस्ड अप्रोच जो है, स्ट्रक्चर्ड अप्रोच जो है, वो इसीलिए इम्पोर्टेंट है कि आपका जो रिस्क है प्रोडक्ट फेल्यूर का वो कम करता है। ओके, so systemic trials, taking systemic trials is a smart, I say it as a smart work, and smart work done by only lazy persons. Okay, so I request all to be a lazy and do a smart work. ओके, सो क्या होता है कि बायो नॉन इक्विवेलेंट अगर हो गया, बायो इक्विवेलेंसी अगर फेल हो गया, तो आपका रीडेवलपमेंट में चला जाता है, आपको फिर से डेवलपमेंट करना पड़ता है। अगर आपको कॉन्फिडेंट है कि अभी ये बायो पास हो जाएगा, तो फिर आपको बायो स्टडी करना रहता है। अगर आपका बायो इक्विवेलेंट पास हो गया, तो आपको क्या करना है, आपको डाटा कलेक्ट करना है, स्टेबिलिटी डाटा कलेक्ट करना है, आपका प्रोडक्ट डेवलपमेंट रिपोर्ट का डाटा कलेक्ट करना है, जो भी इनिशियल डेवलपमेंट स्टडी किया है वो डाटा आपको कलेक्ट करना है, और वो सभी डाटा कलेक्ट करके आपको रेगुलेटरी सबमिशन करना है। एंड रेगुलेटरी एजेंसी जो है वो रिव्यू करेंगी और आपको अप्रूवल दे देंगी। व्हाट इज नेक्स्ट स्टेज आफ्टर दिस अप्रूवल इज स्केल अप। ओके, प्रोसेस वैलिडेट करना और स्केल अप करना। सो up to this point is a second part of my presentation is development, and what is the next is the post development. Okay, पहला हमने देखा prior to development, फिर development, and now it is the post development. तो post development कब होता है, जब आपका प्रोडक्ट अप्रूवल आ गया, उसके बाद में जो challenges है that I have covered in this slide, post development. So beyond the successful bioequivalence study, the challenges are the product approval. One is product approval. प्रोडक्ट अप्रूवल भी में पोस्ट डेवलपमेंट में ही गिना है, क्योंकि डेवलप तो हो गया, फार्मूलेशन साइंटिस्ट टास्क कंप्लीटेड। ओके, साइंटिस्ट क्या बोलेगा कि आई हैव डेवलप्ड द प्रोडक्ट एंड आई हैव प्रूव्ड इट इज बायो इक्विवेलेंट। ओके, ये दिस इज द ऑब्जेक्टिव ऑफ फार्मूलेशन साइंटिस्ट। जो भी R&D पर्सन है, R&D हेड है, उसका रिस्पॉन्सिबिलिटी ये है। बट बियॉन्ड दिस रिस्पॉन्सिबिलिटी, वेरी स्मॉल रिस्पॉन्सिबिलिटी इज दैट टू रिस्पॉन्ड टू द रेगुलेटरी क्यूरीज। ओके, जो रेगुलेटरी क्यूरीज होती है वो बहुत डेंजरस होती है। रेगुलेटरी अथॉरिटी ऐसे ऐसे क्वेश्चंस आस्क कर पूछती है, आप जब हम उस डाटा सबमिट करते हैं तब, तो उसको रिस्पॉन्ड करना बहुत बड़ा चैलेंज हो जाता है। इफ यू फॉलो अ सिस्टमैटिक अप्रोच, देन इट विल बी हेल्पफुल। ओके, तो ये रेगुलेटरी क्यूरीज को रिस्पॉन्ड करना बहुत ईजी हो जाता है। तो मैं ये एक एज अ केस स्टडी में आपको ये भी एक बताऊंगा कि कैसे रेगुलेटरी क्यूरीज आती है, और कैसे अगर आपने डाटा अगर डेवलपमेंट ट्रायल्स आपने अगर अच्छे से नहीं किया है, सो हाउ इट इज़ बैडली इम्पैक्टिंग टू द प्रोडक्ट अप्रूवल प्रोसेस, दैट आई विल ऑल्सो एक्सप्लेन यू इन अ केस स्टडी सेशन. You need to be very well organized, very well structured. Okay, so successful generic product हम किसको बोलेंगे? What we will say, what is the definition of robust product? Robust product हम किसको बोलेंगे? So robust product is what that can be manufactured by any qualified person on any qualified piece of equipment in any qualified facility. बराबर? तो and throughout the years and throughout the life cycle. थ्रूआउट द लाइफ साइकिल अगर आप उसको सक्सेसफुली मैन्युफैक्चर कर सकते हैं, तो ही वो सक्सेसफुल जेनरिक प्रोडक्ट है. सो वी कैन से वी हैव डेवलप्ड सक्सेसफुल जेनरिक प्रोडक्ट. ओके, सो robustness of developed product is a big issue nowadays, and to have the robust product, to develop the robust product, you need to use the QBD principles. You need to understand the ICS Q8 guidelines. Okay, so ICS Q8 guidelines, which tells you about the product development, and it enlists all the process of product development, what to be followed and what not to be followed. कैसे आपको एस्टेब्लिश करना है आपके मटेरियल प्रॉपर्टीज को और आपके जो क्रिटिकल क्वालिटी एट्रीब्यूट्स है उसको. ओके, So spending more time, effort, and planning during the development of any product will certainly generate the robust product. मतलब क्या कि आपको ज़्यादा से ज़्यादा टाइम स्पेंड करना है. So ज़्यादा से ज़्यादा टाइम स्पेंड करना है मतलब क्या? वेल ऑर्गेनाइज. टाइम मस्ट बी अ वेल ऑर्गेनाइज. ओके, अच्छा इफर्ट लेना है, साइंटिफिक वे से आपको जाना है, अच्छे से प्लान करना है और फिर प्रोडक्ट डेवलप करना है. तो इससे क्या होगा कि आपका रबस्ट फार्मूला डेवलप होगा. So in all slides what I explained to you is that overall process of generic drug development and what is the role of formulation development scientists in that, how what quality scientists must have, and what is the importance of this ICH-Q8, what is the impact of this ICH-Q8 on your product development. Okay, and last slide I have told you what is the basis for the successful validation study, means post scale up studies. Okay, so this point I will consider as an case study in later sessions. Okay, post development जो challenges रहता है successful process validation studies का कैसे करेंगे और उसमें रेगुलेटरी क्यूरीज कैसे आती है, वो ये सब हम केस स्टडीज में वन बाय वन हम ये पॉइंट्स हम करेंगे. ओके, सो अप टू दिस एवरीवन इज क्लियर, एंड इफ यू हैव क्वेश्चन प्लीज raise your hand, ask the questions. We will have a question answer session, and after that we will take a 10-minute break, 5-minute break. Okay madam.
हो चालेल, 5 मिनिटाचा ब्रेक घेऊ. If anybody has any question, please raise the hand.
Yes, sir.
Please ask the question.
Please unmute and ask the question.
Good afternoon, sir.
Yes, please.
So my question is, what are the steps in identification of a branded product to convert it into a generic one?
Means you want to say what is the basis for product selection, right?
Yes, sir.
Yes, very genuine. If it is a company and it is, as I already told you, the company is business organizations, the major criteria for product selection is the business. कितना फायदा होगा? See, the first benefit, cost benefit ratio is the first criteria. Okay, ऐसा नहीं है कि मार्केट में क्या रिक्वायरमेंट है, मार्केट में क्या रिक्वायरमेंट है वो डिलीवर करते हैं जिसमें हमको ज़्यादा प्रॉफिट है. ओके, सो फॉर दैट बिजनेस डेवलपमेंट BMO करके होता है, बिजनेस BDO, बिजनेस डेवलपमेंट ऑर्गेनाइजेशन. तो वो क्या करता है, सेल्स फोरकास्ट देखता है. वो क्या करेगा कि जेनरिक जो मार्केट में है वो क्या पहले 1-2 साल 5 साल तक उसको स्टडी करेगा, कितना बिक रहा है एंड कौन से सिचुएशन में उसका सेल्स कैसा था. ये सब वो सेल्स पर्सन जो होते हैं, फाइनेंस पर्सन जो होते हैं, उसको स्टडी करते हैं, एंड बेस्ड ऑन दैट दे कैन डिटरमाइन द सेल फोरकास्ट. वो फोरकास्ट निकालते हैं कि ये जेनरिक अगर हमने डेवलप किया, इतने टाइम पे हमने अगर लॉन्च किया तो ये इतना प्रॉफिट हमको देगा, इतना ये सेल्स हो सकता है. और साथ में ही वो हम ये भी चेक करते हैं कि हम कितना मैन्युफैक्चर कर सकते हैं. ओके, तो जैसे कि मैंने बताया था कि प्रोडक्ट जो डिज़ाइन स्ट्रेटेजी नोट है उसमें पॉइंट्स ये भी आते हैं कि आपका कमर्शियल बैच साइज कितना रहेगा. कमर्शियल बैच साइज में आपको कंसीडर करना है. बेस्ड ऑन दैट यू हैज टू कंसीडर द बैच फॉर एग्जिबिट बैचेस. ओके, तो एग्जिबिट में कितना बैच लेने का है वो भी आप उसके बेसिस पे कंसीडर करते हैं. सो बेसिस फॉर द प्रोडक्ट सिलेक्शन इज व्हाट इट इज अ ब्रांडेड सेल्स फोरकास्ट एंड जेनरिक सेल फोरकास्ट, मार्केट रिक्वायरमेंट, मार्केट में क्या रिक्वायरमेंट आएगा, स्ट्रैटेजी क्या रहेगा आगे, वो बेसिस पे करते हैं. और उसके बेसिस पे एक कॉस्ट बेनिफिट रेशियो निकालते हैं कि हमको कितना कॉस्ट रहेगा, कितना बेनिफिट रहेगा, और कितना जल्दी हम डिलीवर किया तो कितना इंक्रीज इन प्रॉफिट होगा, ये भी एक डिटरमाइन होता है. तो उसके बेसिस पे ये होता है. So इसमें project management is also involved, and business development organization is also involved. Okay, you got?
Yes, sir. Thank you, sir.
Okay. So anyone?
Okay, Mahesh, we'll take 5 minutes break and then we'll continue.
Okay.